We talked about DNA being a store of information and making protein. Why does DNA store information? Because it wants to be able to transfer that information to create an offspring. So DNA replicates to make new cells. So those cells are ready for your body or for the preparation of a new offspring. In addition, the code of information in DNA is actually a message that says, hey, I'm going to make this protein, okay? So we're going to we talked about replication yesterday, and we're going to reinforce that today a little bit. We're going to talk about protein synthesis today, or we're going to introduce, okay? So we talked about the making of proteins a long time ago in Unit 3 when you guys did your cells. What organelle in a cell makes proteins? Who could remember that?
Rough ER.
Rough ER has something on it that makes proteins. Good. So DNA with ribosomes. Oh, I meant here. Let me see. DNA with the help of ribosomes makes protein. Yes? But DNA, in order to make protein, first makes something, okay? And we call the making of protein synthesis. So the first thing DNA does, what is DNA doing here? Any idea? This is deoxyribose nucleic acid. Anybody know what this is? Good job. DNA actually is a template of information to make RNA. DNA with RNA and the help of ribosomes make protein. So first DNA makes RNA, then RNA with the help of ribosomes makes the protein, okay? You could say DNA's job is to make protein because at the end of the day, DNA houses the code that's transferred to RNA that goes to a ribosome, and all together they make protein. And we're going to learn that process, which is called protein synthesis. Since RNA is responsible with DNA and ribosomes for making protein, let's learn a little bit of RNA. And the best way to do that is to compare DNA to RNA. Okay, so RNA, what do you think it's called? Ribonucleic acid. So deoxyribonucleic acid, RNA is ribonucleic acid. Now the left side is going to be everything about DNA, and the right side is going to be everything about RNA. So DNA has how many strands? If you look at DNA, there's nucleotides on this side and there's nucleotides on this side. Those would be your strands. How many? Two. DNA is double-stranded. Let's underline the word D for DNA, D for double. How about RNA? Single. If you look at it, you see it's one-sided. It doesn't have two sides. So it's also built by nucleotides because they're both built by nucleotides, but DNA attaches from one side to another through hydrogen bonds and nitrogenous base pairs. RNA just has one side, okay? DNA sugar, what is it? 5-carbon sugar called D for DNA. D for what's the sugar name? Deoxyribose. D for DNA. D for deoxyribose. RNA sugar is ribose. No deoxy. No di. Just one. DNA and bases. Who can name the bases of DNA or the nitrogenous bases? If you don't know the names, tell me the letters. C.
T.
Let's C always goes with?
G.
G. And then you got T, which always goes with? Good job. So we got A, T, C, G, or C, G, A, T, whatever. These line up, these line up. RNA has a little bit of a difference. Anybody know what the difference is? They have, instead of a T, they have something. A U. So RNA has CGAU, uracil instead of thymine. RNA, C lines up with G sometimes, but that's not RNA because RNA is single-stranded. But they're still attracted to each other. So that's how come sometimes you have DNA, sometimes you have RNA. DNA stays in the nucleus forever. RNA can leave the nucleus. Where do you think RNA is going to go when it leaves the nucleus? It carries the, good job. It's going to go to the ribosome because it carries the information to make protein, and the ribosome is going to help it. So DNA houses information in its nucleotides, and it's going to create an RNA which holds all these messages, and then it's going to leave the nucleus to go make this happen. So we're going to look at how that happens. Just step 1 today, okay? Actually, maybe not. Let's look at the functions of RNA. So RNA is RNA. It doesn't matter what I'm talking about here. But there's 3 functions to RNA, and they name RNA based on their functions. So mRNA is called messenger RNA because it carries the what? The message to do what? Make protein. It literally carries a code, and that code says, bring me this amino acid because I'm going to make this protein. tRNA is called transfer RNA. What do you think it does? Transfers what? Amino acids, right? Because it takes, remember, amino acids are the building blocks. So I need amino acid, amino acid, amino acid, and then I'm going to end up making a protein. So tRNA, oh, this is wrong. Oh, that says R. Should have kept my glasses on. tRNA brings in the amino acid to mRNA to form a protein, okay? And rRNA is just considered ribosomal RNA because it's found on the ribosomes. That's all you have to know. They're all RNA. Their name changes based on their function. All right, so protein synthesis. Step 1 is called transcription. It happens where? Look at the diagram. And that circle is a nucleus. It happens inside the nucleus. Translation happens where? Outside. So it happens here. Remember that DNA is inside the nucleus. It makes RNA. RNA leaves to where the ribosome is, and they finish making protein outside of the nucleus. Transcription happens inside the nucleus. It should say happens. Let's fix that. Happens. And then fix that one too. So let's look at the step-by-step process of transcription, okay? It's going to be similar to replication's beginning, but it's completely different, okay? Remember that DNA does 2 things. DNA replicates in order to transfer genetic material and codes to other cells. DNA's code is also used to make protein. So we're talking about DNA's second job now. So transcription, have you ever heard of something being transcribed? Where have you heard that word? Anybody? Who's a transcriber? A transcriber will write or decode a message for who? Anybody know who a transcriber is? So have you ever seen in court, there's a person typing and typing and typing and typing? So they're typing a weird language that they learned to keep up with the speed of how fast people are talking. It's literally a specific machine where they type information, and then they have to transcribe all of that information and type real words that reiterate everything that was said in court. DNA actually has a message that's going to be decoded during protein synthesis. So the first thing that happens, now look at what's happening to DNA in this image. It's splitting. So the first thing that happens, just like in replication, DNA unzips. Then what happens? What do you see happening? By the way, my DNA model is blue. And what do you think this pink thing is? RNA. Why do you think that's RNA? There's 2 reasons why I know that's RNA. It has U's and? Okay, let's pretend it didn't. Thank you. It has a U and it's single-stranded. That's how I know it's RNA, sorry. DNA is double, okay? So RNA is made, and we're calling it messenger RNA because it's the one that holds the message. And it's using DNA as a template. So if DNA's template is ATCGA, who could tell me what RNA would be? It's going to bring in corresponding, because remember those bases are attracted. Go. Awesome. AU. No, look. AU. What's next? That's a C. So what's next? Okay, we got AU. Who could finish it? I'm sorry.
UA.
It's UA, because A would go with T, right? And instead of T's, RNA has a U. T would go with A, right? So what's C going to go with? G. What's G going to go with? C. What's A going to go with? U.
UAGC.
So it's corresponding bases. I got to remember to keep my glasses on when I'm teaching you so I don't say yes to something that's not there. I do this, okay? When I'm going to make an RNA, I do the corresponding bases of DNA. So I'll do A with T, T with A, right? And then wherever there's a T, I erase it and make it a U. And the reason I tell you to do that is because sometimes you're going to put a U with the T instead of the A. So go ahead and do the corresponding bases you know. Erase all your T's and make them U's. It's a one extra step, but it'll avoid any errors. Then mRNA is modified, so a couple of enzymes and other things come in and kind of fix any mistakes. Remember when we saw DNA replication, there could be mistakes. And what's going to happen to our mRNA? What do you see happening in this diagram? So here it unzips. The new nucleotides come in, which are the corresponding to DNA. And what's happening to RNA right here? Where's it going?
Out.
It's leaving what? The nucleus. It's going to leave the nucleus. All of these three steps happen during transcription.